Hello, good morning. This is um, the second week of our course, Spring Wildflowers, with code BPSC031, lecture one of the second week, corresponding to Monday, April the 6th of the year 2020. Today, we're going to discuss plant vegetative morphology, a few odd bits and pieces that remain to discuss and analyze on the shape of leaves, and in particular on the differences between two large botanical groups, the monocotyledons and the dicotyledons. And then if we have time, we'll discuss a little bit, and we'll start at least with the analysis on the evolution of the flower, the floral morphology of plants, or also the reproductive morphology of plants. Let me start with this plant here. This is a garden plant in the lily family. It has what looks like a stem that gets into the ground. This one broke, but if you go deep enough, you will see that that quote-unquote stem reaches eventually a bulb, pretty much like that of the onion. And the bulb, as we saw it in class, is just a very shortened stem. This is a rosette of leaves. The base of what comes out, what looks like a stem, is really the petioles of the leaves, so also known in this group of plants as the sheaths. So really the stem is down here, the sheaths come out, and then once they reach above ground, the leaves start opening. You will see that in lilies, like in grasses and in many other plants that we're going to discuss now, the leaves are parallel. They're basically long, narrow leaves with parallel venation. You can tell that the venation is parallel because if you pull them apart, they break longitudinally. They form the conductive tissue, the veins, what conducts nutrients into the leaf, are arranged along the blade of the leaf, and they go all in parallel form all along the blade of the leaf. This is quite interesting. There are many plants that have parallel venation. And Linnaeus, who was an acute observer of nature, realized that parallel venation in leaves came together with a number of other traits that were indicators of two large groups of plants that were quite distinct. Plants with parallel venation are normally herbaceous, as you can see here. They don't have any woody tissue. They don't produce large trees. You will never see a large tree with parallel venation with leaves like this. They basically also have flowers in groups of three. They have three sepals, three petals. We'll see later what that is. But the flower parts are in multiples of three. And if you germinate the seed and look at the seedling after germination, you will see the first leaf in a seedling when a plant germinates is known as a cotyledon. And you will see that the cotyledon in these plants with parallel venation is only one. It only has one cotyledon, while the plants with reticulate leaves, with normal, quote unquote, normal leaves, have two cotyledons and flowers in groups of 4 or 5. And if they live for more than 2 years, they will produce wood. They produce trunks with wood and growth rings. They will grow every year and leave the dead wood inside the trunk, and they're producers of wood. So there are a number of plants, some of them economically very, very important, that belong to this group that are known as the monocotyledons, plants that have only one cotyledon. Let me show you a few. Grasses, for example, have leaf blades that have parallel venation. And this is something, grasses are so ubiquitous, so common, that you will immediately recognize the leaf blade of a grass. We'll see the grass family later on. But yes, they are in the family, sorry, in the group, in the lineage of monocotyledons, like daisies, like many other plants, incredibly important. most of the calories we eat, what keeps humanity alive, are grasses, a few grasses like rice, corn, wheat, barley, rye, or oats. Basically, grasses are what keep us alive. Incredibly important, and with parallel venation monocotyledons. The Joshua tree, known in botany as yucca, Yucca brevifolia. I didn't kill a Joshua tree to get this for you. There was a neighbor that was clearing some branches from a Joshua tree, and I got a branch for you. But look at the leaves. The leaves don't have any reticulate venation. Let me show it to you. The veins are parallel, and you can tell immediately because if you pull them apart, you see they break longitudinally, right? They have veins that separate longitudinally. Let me show you here again. The veins separate longitudinally. Perhaps more complicated to interpret, but I hope you will finally agree with me, is this beautiful plant that you see in many gardens in California, the bird of paradise, whose leaves look pretty much like the banana leaves also. And this one, when you see it, say, hey, this looks like a typical leaf, doesn't look at all like a grade. It has a petiole, it has a midrib vein, a central vein, and it has lateral veins. Yes, but look at the lateral veins. The lateral veins are all parallel distributed. You can break it. I'll try and break it from the side, and you can see how it breaks parallel to each other. So again, although the central vein, all the rest of the venation radiating or from that central vein is formed by parallel veins, also belongs in the monocotyledons. Finally, the most interesting one perhaps is this very complex leaf here, the palm leaf. This is the California fan palm. And you say, okay, that is a palmate leaf. It has nothing to do with parallel venation. There's nothing parallel in those folioles, those leaflets radiating from a central point. Yes, but sometimes it pays to look at leaves when the plant is very small, when the plant is a seedling. And if you look at these seeds when the plant is a seedling, what you will find is something like that. It looks very much like a grass. And if you look at it even smaller than this, it's almost undistinguishable from a grass blade. And then, as it starts growing, it will start becoming wider. And it's almost like an accordion. It's pleated like a Scottish quilt. And it becomes wider and eventually starts opening up here on top. You will see one here. It starts opening there on top, sort of flecking out until it actually forms something that looks pretty much like the leaf of a palm. And you can imagine as it grows, as the leaves become older and older, they become more this trait in which the original parallel veined assemblage of the leaflets become wider and wider until it forms the typical fan of a palm leaf. So here I have a summary of the development during the lifetime of a palm, of the typical palm leaf. When it germinates, this is the seed, one single cotyledon that attaches the seed to the seedling, and the first leaf looks almost indistinguishable from a corn seedling, for example. It looks very, very similar to any seedling in the grass family. Then as the leaves grow, they start becoming more and more pleated and somewhat wider until they start sort of separating on top into these dented apices. And then, of course, they start separating more and more and opening radially until they form the adult leaf of the palm. But at the end of the day, if you look at the leaflets that form the leaf of the palm, let me get one leaflet here. If you look at the leaflets, each leaflet looks pretty much like the rest of the group in the monocotyledons. It looks very much like a grass blade. You look at this and you wouldn't distinguish it from the blade of a bamboo plant, for example. It looks very, very much like a grass blade, and they separate longitudinally because all their fibers, which are the conductive tissue that runs along the leaf, is organized in parallel veins. This is in total contrast, with the dicotyledon leaves, and I'm going to show you a few just to get an idea and an understanding of what's going on. Let me show you two typical dicotyledon leaves. First of all, the macadamia nut. You can see the leaf. It has a central vein that runs all along the length of the leaf. As we studied before, the upper surface is green and bright. The lower surface is duller because it's mostly used for gas exchange. And if you look at the venation, you will see that it's reticular. It has the midvein and then primary veins that separate into secondary veins that separate into tertiary veins, and so on and so forth. So dicotyledons have really reticulate venation. Some of them start from the very base. You can see this is a leaf. It's a somewhat crummy leaf, really, but anyway, it's a leaf of a fig tree, and you can see from the base of the pistil one vein coming here, another vein coming here, another vein coming here, and a smaller vein coming in this direction. Each of them radiating into second and third order veins. So basically, this plant doesn't have a primary vein. Everything radiates from the base of the leaf or from the apex of the pistil, but it also has a reticular formation of. Let me put this into an image so we can get some systemic idea of what we have been discussing. The leaves in monocotyledons are mostly parallel veined, as in the grasses, the lilies, or they're pinnately veined, in which case, as in the bird of paradise, they have one central axis and then parallel veins radiate or branch off from that central axis. This is a typical example also of the banana leaf, one central vein with only first order veins parallel to each other radiating from the central axis. While in dicotyledons, you either have reticular venation, that we saw for the case of the macadamia nut tree, or palmate venation, which is similar to, we saw that for the fig. It's similar to the reticular venation, but they all the veins radiate from a central point in the base of the leaf. So basically, summarizing the whole idea, leaf venation in monocotyledon species is mostly parallel, and it can be pinnate. That is, it can have a central axis and then radiating from there. In dicotyledon species, venation is always reticulate, and it can radiate from a longitudinal vein, in which case we call it circular reticulate, or palmate, in which case veins radiate from the base of the leaf or from the apex of the pistil from below into different first order veins, and no mid vein is present. Leaves can be classified according to a number of traits. One easy one is the overall shape, how wide it is in relation to the length. If the leaf is like a pine needle, it's called acicular. The leaf is needle-like. It's very, very narrow in relation to the total length. If the leaf is elongated and with parallel venation, several times longer than wide, and the sides themselves are parallel, then the leaf is linear, like in grasses or in lilies. If the leaf is widest at the middle, it can be elliptic or oval, and we'll see the difference in a second. And if the leaf is almost perfectly rounded or circular or nearly so, it's called orbicular. Here we have three illustrations that exemplify the shape, and we are going to see some here from samples I got. For example, the leaf of this tree called Feijoa selwayana, the false guava, is perfectly orbicular. It makes like an orbit, right? It's wider in the middle, around half of the length, and it resembles like the orbit of a planet. It's called orbicular. The leaf of the orange tree on the other side is oval. It has it's wider it's longer than wider. It's quite wide, but the widest part is below. It's more elongated on top than in the bottom. So it's not. It's not. It's oval. Sorry, I'm being silly now here. One interesting leaf is this one, the leaf of the nasturtium. This one, as you see, the petiole is not inserted in the bottom of the leaf, but it's inserted in the middle. And so the leaf is orbicular. It's almost perfectly rounded. It's almost a perfect circle with the petiole inserted in the middle, and the venation is palmate. It radiates from the center of the leaf all around it. Leaves can be classified by their overall shape also. Like, for example, if a leaf is triangular, we call it deltoid or deltate. If the leaf is like a spear, we call it sagittate. Let me show you some illustrations because I don't have enough leaves to show you. Let me share the screen here. If a leaf is like a spear, is lanceolate. If a spear is like an egg, is ovate. If a leaf is like an egg, it's ovate. If a leaf is like the top of an arrow, like the tip of an arrow, it's sagittate. And if a leaf is like a twisted spear, we call it falcate. Falcus in Latin is a sickle, the tool that was used to harvest wheat. You can classify leaves by the leaf margin. It can be entire, it can be sinuate if it has like a sign function around it. It can be crenate if it has acute enterings. It can be dentate if the acute tips are pointing outside, and serrate if it looks like a saw. I don't want to spend too much time in the nomenclature. It can be complicated, but just draw to your attention that in your lecture notes, you have in good detail all the nomenclature of leaf parts. And so you have to describe a leaf. It would help a lot if you just look at the lecture notes, look at the leaf parts, and describe the leaf according to the nomenclature that you have there. Now, we're going to move on now to the second part of this class, sorry, which is the origin of flowers. Let me load the PowerPoint images and we'll talk in a second. Okay, here we are. In order to start talking about the reproductive morphology of plants, let me start with a little story that will lead us into what we want to discuss. Some years ago, there was a student from the University of Mexico that came to Riverside on an education abroad grant and spent a whole year taking courses here in Riverside. He took a number of courses on botany and also the course on vegetation in California with Professor Richard Minnick, which is a wonderful course. And he became very much in love with the history of vegetation in large ecosystems. And he traveled the world. There's a photo of him in Peru, in Machu Picchu. And from once he finished his bachelor's degree in the University of Mexico, he got a grant to do his PhD in the University of London in England. And he did a wonderful project that led to the publication of his thesis in the form of a very, very influential paper, a paper that made really a big splash in the scientific environment, that was called Cretaceous Forest Composition, inferred from a global fossil wood database. But basically what he did was he got a fellowship in England to travel different museums in Europe in general, and look in the museums at the fossil collection of plants. And that sounds like a dream PhD program. Or project. He basically toured the museums in Europe, seeing collection of fossil plants from all over the world, because European countries have been colonial countries that have had colonies all over the world, very often dreadfully managed for the people in the colonies, but whatever it was, they have fantastic material from all over the world, from Siberia, from Mongolia, from tropical Africa, from South America, in those museums. So he went to Berlin, to Madrid, to Rome, to Paris, to different museums in England, in Edinburgh, in London, and he asked for the boxes with plant material from a period known as the Cretaceous, and in particular around 120 million, dated around 120 million years ago, and checked what was the dominant vegetation that fossilized in the earth around that time. And he found that the earth 120 million years ago was dominated by araucarias that are known as monkey puzzles, podocarp trees, which are also in the conifers, cypresses, and pines. So some 120 million years ago, the earth was covered basically by conifers. All ecosystems were covered by conifer plants. And then 65 million years ago, after an event that was called Chicxulub, and I'm going to tell you in a second what that event was, suddenly the earth was covered by magnolia-like flowering trees. So some 65 million years ago, the earth changed. And it changed, as we will see, in its composition, its botanical composition. It changed rather abruptly. Emiliano's findings made a big splash in the scientific environment, and also many newspapers in Europe. He was all over the place in his articles in newspapers that were using his data to try and reconstruct how the earth must have looked like 120 million years ago, drawing these funny dinosaurs and a vegetation of what we know as gymnosperms, which is basically conifers and cycads. And basically what Emiliano showed is that 120 million years ago, the earth looked pretty much like this. This is a photo of an araucaria forest in Chile. And araucarias are known as monkey puzzles. They are very interesting trees. They have changed very little in the last 200 million years. They have been pretty much the same for around 200 million years. They have changed very, very little. Now they only grow in very few restricted places on Earth. They are, like we would call them, quote unquote, living fossils. But at one point, as Emiliano demonstrated, they were covering all over the world. And they were the main tree that coexisted with the dinosaurs. Emiliano also showed, and many other researchers have showed, that the transition from conifers to magnolias was really very easy to understand. Conifers, as the name suggests, the reproductive organ of of, uh, of conifers is basically this structure here called a cone. And a cone is a shortened stem, like a rosette. Remember when we studied rosettes, a very shortened stem where the leaves are somewhat transformed, and instead of being green and photosynthetic, they're mostly flat and lignified, woody. And inside the leaves, you have organs that bear in these leaves. Inside these leaves are known as bracts, B-R-A-C-T, which is the name for a simplified transformed leaves. Inside these bracts, there are either pollen-bearing structures or ovules that are fertilized by the pollen. They're fertilized by wind. They have no nectar, they have no pollinators, they just produce a pile of pollen into the air, and some of the pollen will finally make it to another cone that is a female cone and fertilize it. If you look at a magnolia, as a matter of fact, which is one of the earliest flowering plants, as Emiliano demonstrated, they were around 65 million years ago. They were the first flowering plants to be around on Earth. You will see that it has also a stem, a shortened stem, like a rosette, but the bracts are much wider and creamy and sort of appealing in more than one way. And they will produce, many of them will produce some nectar at the base. But the basic structure of the cone is still there. You have a series of transformed leaves that are petals, and then you have a series of transformed leaves here in the center. You can see they're wide but much smaller, that have anthers, pollen-bearing structures at their tip. And then you have a series of transformed leaves up here that are basically flat structures close on each other with an ovule inside. So there's really not that much difference between the cone and the magnolia flower. The magnolia flower is like a specialist cone where the bracts have evolved to play different roles. As a matter of fact, recently there was an article in the journal Nature where a group of researchers reconstructed how the first angiosperm flower, how the first flowers must have looked based on genetic analysis of genes common to all flowering plants, and working backwards on very ancient genes that have existed on flowering plants since the origin of flowering plants, and their inference of what an original flower must have looked. Look at it and compare it with a current magnolia is not that different. Magnolias have also changed, like araucarias, have also changed very little in 65 million years. So what happened 65 million years ago that suddenly led the world to start having plants like the magnolias, plants with flowers, with very rudimentary flowers still, occupy the world at the expense of conifers that became only restricted to some very particular ecosystems? Well, what happened was this. A huge comet came and hit the Earth some 65 million years ago. It was so big that it didn't melt, it didn't burn in the atmosphere, and it hit the Earth with a power of around 1,000 atomic bombs, or thousands of atomic bombs. I don't know how many. I don't even think that anybody knows exactly, but basically it hit the Earth with immense power and produced a crater that is hundreds of miles in diameter and is still can be detected under the corals and the water of the Yucatan Peninsula. It's buried now, but it's still there, and it's being studied currently as we speak by geologists. It's one of the hot topics of research on Earth history. And basically the explosion of this comet, known as Chicxulub, following the name of the Maya town where it was first discovered, the explosion of the Chicxulub comet released so much energy, equivalent to thousands of atomic bombs, that it covered the Earth with the explosion dust. And it was like a nuclear winter that lasted for at least 10 years. It lasted so much that in the geologic record, there is this layer known as a KT transition line that appears in every sedimentary rock formation, and it's full of radioactive iridium, indicating that really the Earth went through a period of around 10, 15, 20 years of high radioactivity in the atmosphere, and the atmosphere all over the world. The KT transition layer appears all over the world, and that must have really put the Earth in a mode of very low productivity, like a nuclear winter. Photosynthesis collapsed. Plants did not have enough light to do their photosynthesis and their growth, and only a few plants survived and a few animals survived, mostly small animals. All the large animals, the dinosaurs, went into extinction after the Chicxulub explosion. And only a small amount of animals was able to survive, mostly small reptiles that are still around. We see them in the form of lizards and things like that, tortoises, birds that are also still around, and a group of very small animals that were warm-blooded and had hairs, but they were rare and a minority in the richness of animals in the Cretaceous. And these were the ancestors of mammals. They were warm-blooded and they gave birth to live offspring. They could rear the offspring inside the womb, and they were mammals. So these animals, the mammals, and of course the insects. Insects survive in huge numbers. These small animals, mammals, birds, insects, reptiles, colonized the Earth, and in particular, the magnolias that existed before the explosion, and a few managed to survive, started being attractive to insects in the middle of a very dreary environment after the explosion, because they were producing some sugary liquids from secretions in the flower. Little glands in the flower whose origin is not very well known, probably was only just a result of the cells of the development of cells at the base of the bracts that got easily broken. And insects started visiting the flowers because the bracts were so soft and they were nice to munch upon. And suddenly the plants that had showy bracts attracted more insects, and the insects would take the pollen from one plant to another in their visits to different flowers, and the revolution of flowering plants occurred. In geologic terms, it was incredibly fast. In a couple of million years, it might seem a lot to us nowadays, but it's a very, very short time in evolutionary time. The flora of the Earth made a huge transition from plants with no flower, basically conifers, to plants that have attractive bracts instead of having woody bracts like conifers, that could attract pollinators and compete for the service of sending pollen to other plants. The advantages of pollination by animals was huge, and the advantages of having flowers that could aid in pollination was immense, because the flowers could actually produce much less pollen. Really, conifers have to produce huge showers of pollen in order to have some of that pollen reach through the wind a female cone. Here it could be directed very precisely to the next flower by the simple act of giving the pollinator a little reward in the form of sugary secretions from the sap, in the form of plant sap. And so the origin of flowering plants started very soon, and it radiated into an explosion of life forms and families some 65 million years ago, which is the most currently accepted date for the origin of angiosperms. So what we will see in the rest of these classes, until we get into the specific families, is the morphology of the angiosperm flower, how those flowers evolved, and how they operate and how they act in terms of plant reproduction. We'll start this talk next class on Wednesday, and I thank you very, very much for your attention, and I wish you a great 2 days until our next class, which is Wednesday the 8th, week 2, lecture 2. Thank you very much.